Hi, everybody. Welcome to another Chats with Chaudhrey. I hope you're safe and well today. I'm delighted to be joined by Jamie Tempest, who's Senior Scientist 1, and Thomas Gosling, who is Scientist 2, from Rssl, which is Reading Scientific Services Limited. And we're going to be talking about sterility testing today. So, Jamie, Thomas, it's lovely to see you. How are you both?
Yeah, I'm good, thanks. Yeah, not too bad at all.
Yeah, very good,
Fantastic.
thanks.
Great. And Jamie, where are you calling in from?
So just outside Reading, just in Shinfield, working from home today. So I didn't have the
Cool.
cold commute.
And Thomas, I understand you're in the office.
Yes, I'm on site today. I had a nice cold cycle to wake me up, just ready for this little chat here.
Yeah, well, it is extremely cold here in Surrey, so I don't envy you getting on a bike and cycling in the frost. But anyway, each to their own. So,
Yeah.
look, before we start talking about sterility testing today, would one of you be kind enough to give me a quick overview of RSSL?
Yeah, sure. So Rssl is a contract testing organisation. So we specialise in pharmaceutical testing and food testing. So myself and Thomas are from the pharmaceutical side. We're pharmaceutical microbiology, but we also cover chemistry, microscopy. We have a training arm as well. So just about anything pharmaceutical related, RSSL can help you with.
Great. And your own background. So, how did you get to be Senior Scientist 1 at RSSL?
So I started off as a scientist one at Rssl, doing my master's degree alongside, well, yeah, alongside work. Yeah, and then the opportunity came up that we were developing a sterility test suite. So there's the opportunity for me to put my master's degree, my engineering, and my sterility knowledge together to help build the suite, and then manage the team that are
Fantastic.
currently in it.
And Thomas, what about you? How did you wind up at RSSL?
So I studied microbiology at Reading University, and then with the RSSL main site being based on the same campus, I'd seen it walking past. So I found a way to be able to start working there. I started doing a wide range of analysis before specializing in endotoxins, and then, like Jamie, once the opportunity to work in the sterile laboratory opened up, I jumped at that chance. Being able to work in a clean room with the isolators, it was a good opportunity to develop into steriles. So, and that's
Fantastic.
been me ever since.
So let's talk about sterility testing. My basic question, first of all, though, is what is sterility testing and why is sterility testing important in the first place?
So I'll start off there, Thomas. So we'll start off with what is sterility. So sterility is the freedom of viable microorganisms. So therefore, sterility is an attribute. Something can either be sterile or non-sterile, and that is essentially what the sterility test is looking for. Which state is that pharmaceutical product in? Yeah, it's a regulatory requirement, so companies are required to do it. Yeah, for both terminally sterilised and aseptically produced products.
Right. Okay. And why is it so important, though?
So the end patient is potentially immunocompromised. You're in hospital. You're then going to receive this drug that's sterile. So the only reason for it to be sterile is it bypassing all your body's natural defenses. So, for example, a parenteral, it's going to go into an IV, into your arm, into your blood. If it's not sterile, it potentially it's a pathogen entering your bloodstream. You're potentially already immunocompromised. It could result in illness or, worst case, death of that
Right.
patient.
Okay. And Thomas, have you got anything to add to that?
Yes, I'll just say it's important to remember the limitations of this test. So it's not on its own going to allow you to declare a product sterile. This is part of a much wider array of tests, everything from in your manufacturing plant with your environmental monitoring and other data. And this, what we provide is an extra set of data along with that to allow you to release the product to market.
Right. Okay. Now let's talk about regulations. Obviously, pharma is a very regulated industry. So, what are the regulations around sterility testing, and how do you, as a business, comply with that?
So regulations generally in the pharmacopoeia. So USB 71, European Pharmacopoeia 2.6.1, and JP 4.06. There's also in the UDRELX Volume 4. There's Annex 1. Which is all the information about not sterility testing. It's about clean rooms, designs the environment you need to then perform the sterility test within. So we have a QAU team that works with myself and Thomas, and every time there's an update to any of those regulations, that's pushed through. And yeah, it's essentially it's one of my as managing the team that in Sterility, it's my job to make sure that we're always in compliance with that.
And I know Annex 1 though has been sort of been muted, has been updated for quite a long time now. So, is there any news on that, or how would that impact what you've done to date with the lab?
So no news. The last piece of news I heard was it was supposed to be out in September 2021, and that obviously didn't happen. So, and in terms of what we did about the draft annex when we started to build the suite, that was all in consideration. So my designs, the equipment we were putting in, the processes and SOPs we put in place, that was all not to comply with the current Annex 1, because that's the current regulation, but it's also to do that one step further and make sure we're already ready for what could be implemented in the next version of Annex 1.
Right. So for those who are not familiar with what a Sorority suite would look like or what it would contain, would you mind giving viewers a sort of an overview of what the suite would contain? I mean, particularly as you built it from scratch, basically. So what does the suite contain, and what are the elements within it that are important?
Sure. So it's a series of rooms, some clean rooms, some non-clean rooms, all designed with contamination control in mind. So keeping the area where you're doing the sterility testing the cleanest. So it's a series of rooms. So we have entrance area, so what we call a controlled not classified area, an incubator room that's controlled not classified as well. That then leads through into a gowning room, which has to be grade D at rest. Then we move through into the clean room, which is grade D in operation, which houses our grade A isolators where we perform the sterility testing. So it was designing pressure flows, cascades, work process flows, material test flows, things like that to ensure minimizing contamination and mitigating it where it does arise.
Right. And Thomas, have you got anything to add to that?
Just to go down to the isolators, they're where we eventually carry out the majority of the work once it's passed through to the final stage there. They're basically enclosure that allows you to separate your products and materials from the surrounding environment. So it's a grade A, basically a grade A clean room that's sanitized with VHP. And this allows the tests to not be compromised, and allows us to release results with complete confidence to the client.
Brilliant. You said VHP. What's a VHP?
So it's vaporized hydrogen peroxide.
Right.
It's used to. It would probably take quite a while if we go into the full.
Right. All right. Fantastic. All right. Now, what are some of the key considerations, though, and things to watch out for when you are actually choosing a sterility service provider?
So anyone that's going to be choosing a provider, you'll be doing a thorough audit of that with Rssl. You want to be making sure that all the controls, as Jamie said, we designed it in mind with contamination control, that those processes work, that they're very well documented, and that people are trained in those processes. You can also go back into the more admin behind as well, and all the media we use, or someone should use, would be QC'd. Making sure it's again viable for use, and making sure that everything is in date, all the kits we use are appropriate as well.
Okay. And Jamie, have you got anything else to add to that? If I'm sort of looking for a provider, what the key things?
Thomas touched on it there a little bit. Training. So, for example, at Rssl, if you want us to be trained specifically in your product type, if you're a unique or different product type, and you want to be sure that we're aseptically handling those, we're happy to do things like aseptic training on those products, supply you with the results so that you're confident, and just be able to basically work with you to help you and make sure there's going to be no stumbling blocks on the way, make sure that everything that we do fits exactly what you need, and things like that. Make sure they're compatible rather than just looking at price is my advice there when you're choosing a new provider.
Right. Okay. Now, can you give me some of the challenges, though, or some of the sort of challenges that you might face when you're carrying out sterility testing?
Sure. So, like with all scientific analysis, you're going to eventually come across some problems, especially when you're dealing with new product types, new materials. So, for example, one we had last year, we were seeing a small amount of growth between 7 and 10 days into the 14-day test. This was seen across a variety of different replicates for this one product. We were unable to culture the growth that well. So we did what we could in order to try and find out what it was. We did our internal moldy tough. We did some external testing as well in order to find out what it was.
Yeah.
This was not noticed during the validation because the validation is finished at 5 days or whenever the growth is seen. So this is before this next stage of potential growth was seen. And then I'll pass on to Jamie, who did a lot of this work, who can fill in the details.
Right.
So touching on where Thomas left off with, so we basically had some growth in a canister that looks like mold. So yeah, our client agreed. We thought that looks like typical mold growth, but we couldn't culture it. So what we did was we took a little bit of that material and we put it in the moldy tough to see if we could just directly ID it. It wasn't successful, but what it did tell us is there were definitely proteins there. So yeah, the client, we organised to send off for rapid sterility. We sent it away, no growth on any of them. So right, so we've got a protein here that's potentially not a microorganism based protein. So we sent it to our microscopy department, who basically did some tests on it and came to the conclusion that it was a protein. Some of the silicon we had from our growth media had passed. So in the rubber bung in the top of the growth media, you put the needle through, there's rubber, and tiny amounts of that had passed through to the filter. Not that you could see it, but yeah, that acted as a nucleation site for the small amount of residue of the protein, the serum that was left on the filter. And yeah, day 7 to 10 caused that turbidity.
Right. Okay. So does that mean then that you have to be careful on using that particular sort of container for future samples?
So we now know that is going to happen in the future. So we have put steps in place. We now have a step additional to the sterility test, is where we actually then go and send it to our microscopy team for them to do the analysis. Basically give us a report to say it's a protein, but it's not microbial. And then we can then go on to release the certificate of analysis for the sterility test.
Right. Well, that leads nicely to my next question, which was really, are there any samples that are a bit of a nightmare to work with, and how do you sort of get around those particular issues?
Yeah, so although the vast majority of our samples are quite nice and simple, to be honest, nice aqueous solutions, there are certainly some which can prove a bit difficult. They might struggle to filter through the membranes, we may struggle to dissolve them, or there might be a medical device that needs to be tested through, say, direct evacuation or something else. We're able to do some feasibility work here at Rssl. This allows us to use a small amount of the client's sample and work out the best method, rather allowing us to try all the kit types, all different media types, and
Right.
potentially anything else we would need on top. This is particularly useful when dealing with, say, medical devices and, as we've seen recently, a lot of antibiotics.
Right. And Jamie, have you got anything to add to that?
So yeah, it's just say that antibiotics and antifungals we've been dealing with recently have definitely been the more challenging ones. Some of the properties have meant that they've absorbed into some of our test materials, so we're not able to wash them away. So then they're leaching back in over time and killing the organisms present in the broth. So yeah, method suitability, method validation, it's been challenging, but this is the sort of thing I like. Yeah, I like it when it doesn't work, and I've got to use some science to try and figure out how we're going to get this to work.
Sure, I can understand that. Does that mean though that because obviously you are testing all these different samples and you are having to do different test methods, that you develop your own unique database or library, if you like, of test methods and how different samples will react to different things, so that you can use for future reference?
Yeah, so yeah, each client's test method is their own, so we don't share client test methods with others, but obviously we have the knowledge, the database is the people, we have the knowledge, we know what's worked, we know what hasn't worked, and yeah, we're able to pass that knowledge on. And because yeah, we do so much of the pharma and biopharma industries, we come across so many different products that you probably wouldn't come across just as you're on your own on the manufacturing site producing your product catalog. We see a lot, and the chances are we've probably seen it, we can probably help.
Fine, brilliant. Well, that actually leads on very nicely actually to my penultimate question, which is, how can Rssl help improve testing methods, and are there any tips you could offer viewers out there who are carrying out sterility testing?
So as a tip, I would say it's always good to get on top of your outdated validations or outstanding ones. This, due to the fact that the test is 14 days long, plus an extra processing time, means that, say you have a problem with your other supplier or other testing laboratory, it will take a long time to get this sorted. If you've got someone set up, got these validations ready to go, it means we can help a lot faster, and meaning no deadlines are missed for your for important drug deliveries. Recently, in terms of what we've been able to do to improve testing methods, we had a client which was using some old Sterility test kits and wasn't using some of the new ones that were available. We were able to then bring in these new kit types. This meant we reduced a step in the procedure, which then reduced contamination, made the testing much easier for us, and as mean we're also able to reduce the cost for the client as we're reducing a whole step for them. So this has helped both us and the clients with just a simple kit change and updating a validation.
Right. And Jamie, anything else to add to that?
So yeah, just to build on what I was just talking about a second ago, really is, yeah, we do so much of the testing that, yeah, the chances are we can help, whether that be the traditional pharmacopoeial test method or whether you're looking at the newer rapid sterility. So yeah, at Rssl are actually in the early stages of onboarding rapid sterility testing. So if anyone out there is watching and they either want to partner with us or want to have an input on which direction we go with it, please feel free to get in touch with me. So.
Brilliant. Well, that leads nicely, I can't get my teeth in this morning, nicely to my final question, which is where can people get more information?
So our website, primarily, yeah, there's all the information on the website. There's also a chat, not bot, there's a person on the other end of the that, or directly contact me or Thomas through LinkedIn or email or give me a call. Brilliant.
Well, gentlemen, thank you very much for talking me through sterility testing. It's really interesting to learn more about that. So there you go, viewers. If you'd like to know more about sterility testing and how Rssl can help you, or if you've got any questions around sterility testing, then please get in touch with Jamie and Thomas. You can leave comments or questions for them below the video. You can message them on LinkedIn directly if you would like to. And also you can go to the Rssl website, and they have got a little person pop up on the bottom right hand corner. As I know, every time I go onto the website, I will ask you if they can help you, so you can ask some questions there as well. And also they've got lots of training as well that they offer, which can help you as well. So all that's to say is, Jamie, Thomas, happy testing for the rest of 2022. I hope you have a fantastic year, and I hope you find some challenges which make it interesting for you as well over the next 12 months. So thank you very much and have a fantastic 2022. And thank you, viewers, for watching. I hope you found that useful. And until next time, as always, stay well and stay safe. Bye-bye.